Thanks, Joe. So I was here last year, and Joe must have loved what I said last year and kept me on the agenda. So here we are. So I'm going to talk about a few things today. One, I'll be discussing our pigweed identification assay or test that we have and some results with that. Continue with our ongoing statewide genetic survey results, upcoming projects, and one of my favorite things to do is beg the audience for samples. So if you haven't heard of us, you weren't here last year, we are really close. We're actually the nosy neighbor for the federal USDA folks and NDSU. So we're housed in the Red Roof USDA building, just kitty corner to the Fargo Dome. We are a not-for-profit private lab, though. We're not associated with either the feds or the state or the university. We're a small group. There's 6 of us full-time that live here in Fargo. Our boss is in St. Louis, and we have 1 intern. We are also ISO accredited. What that means is we pay someone money to come and check our quality assurance and tell us what we're doing wrong and how to fix it. So I'm here to kind of tell you about our DNA testing and how we use it as a multi-tool. There's several things that we can look at when it comes to DNA testing. And our general approach is really to collaborate. We are just a laboratory. We do not have a greenhouse. We don't have test plots. We are here to help with the technology that we have on hand. So we're here to complement, not compete, and really provide confidential reporting to clients. Our clients are university researchers, federal researchers, growers, agronomists, consultants, you name it, you can use our testing. So if we look across the last 9 years, this is kind of 2 categories of our testing. So pest identification and trait identification. Trait identification can be in pests themselves or in the crops. And so if you kind of scan quickly across to last year, most of what we do is pest identification, and the herbicide resistant testing I'm categorizing as trait identification. So we're starting to see some slow growth in that category. We have 36 tests for various crop pathogens, 23 tests for honeybees, 5 genotyping panels for various variety ID in crops that we work closely with the North Dakota State Seed Department with, and then 6 tests for weed herbicide resistance. So really the value of lab diagnostics, you probably don't need to be convinced of this, but even if you can't treat for what we test for, that information helps reduce uncertainty. So it helps you from avoiding chasing the wrong problem. So a great example of that is if it's a bacterial infection in the field and you apply fungicide, it ain't gonna work. And so we can help you decide whether or not to do those type of systems. Another example is if there's herbicide-resistant weeds for a particular herbicide and we confirm it genetically, that allows you to figure out kind of some new integrative weed management strategies. Now, taken all together, if we get enough samples in from the group, we can start associating and looking at patterns of certain pests and pathogens, and also develop kind of an early warning system in those areas. So I'm here to talk about weeds though. So our weed focus approach, like I already hinted to, is to be the nosy neighbor and see what all the weed scientists at NDSU are doing here on the main campus, as well as at the research centers across the state. And so a lot of our work really relies on communicating and helping out what's going on in the greenhouse when it comes to weeds. And really it's to increase access of DNA tech to everyone and also provide fast turnaround. So as I already hinted, when it comes to DNA testing, you can look at a couple things, and I'm going to talk about pigweed identification and then herbicide resistant ID. Let's start with pigweeds. So there's kind of 2 types of samples that we receive from folks when it comes to pigweed identification. The first is confirming Palmer amaranth in new counties, and all that takes is a leaf sample. Secondly, we're looking at the seed sample as well. So we get a lot of amaranth seeds from purity testing from across the US state seed departments. And so the one thing about pigweeds and amaranth in general is all their seeds are these small, black, about 1 millimeter in diameter seeds. And so with our test, we can differentiate several amaranth species. So we can detect if one of those seeds out of those 200 there are Palmer amaranth, or if one of those seeds is water hemp within a background of pigweeds. And so what I mean by this is how the test is kind of set up. So it's a 3-in-1 test. And so the testing, without getting too technical, there's a series of probes, 3 in particular. There's 3 different colors. If Palmer is in that mixed sample of seed, it fluoresces blue. If it's water hemp, it fluoresces green. And if it's pigweed, what I mean by that is if it's redroot pigweed, or if it's smooth, or if it's Palo amaranth, it fluoresces red. And so we can see that mix using light detection or light sensitive equipment. So when it comes to our pigweed identification, last year we got 67 submitters to the lab, 604 total samples. And if you separate it out to the number of small black seeds we tested, it was 21,000 seeds. So the data that I'm going to show you is kind of biased. It's samples that came to the lab. So it's not the distribution of Palmer amaranth in a particular area. It's what we got as a lab. And like I said, pigweed ID is used primarily in the West. So it's a lot of state seed labs, university extension, and Department of Agriculture's that use this test. And so here are the results over the last 7 years that we've been doing this Palmer amaranth identification. And so if you look across the years, I'm just showing you each bar is the total number of samples. So that's the y-axis. The horizontal axis is the year. And so about 1 in every 4 sample that we get in, whether it's a leaf sample or a collection of small black seeds, about 1 in every 4 is Palmer amaranth. And so what are the outcomes of using this test at NAGC? Well, we've confirmed Palmer in new regions, which I talked about. Out west in Oregon and the state of Washington, it's also stopped further spread of Palmer in wild bird seed. So there's been various brands of bird seed that has been spreading Palmer. And so we were able to detect it using our assay, as well as stopped further spread of Palmer in livestock feed. So there are certain places that provide screenings for livestock feed, and if Palmer's in there, it stops the sale for further spread. All right, so that's what we use the pigweed ID test for. Moving on to herbicide resistance. So there's 2 flavors, which was kind of hinted at during the keynote today. There's non-target site and target site resistant mechanisms. So when it comes to non-target site mechanisms, these are enhanced metabolism. It's the way the plant can block the herbicide from reaching its target. As a geneticist, as a person in a lab, it's way too complicated for us to do the testing. So what we're really focusing on are the target site mechanisms. These are simple, generally point mutations in the genome. So if you look at the target site for the herbicide, particularly it's an enzyme, there might be a point mutation along the gene or the genome that changes the shape of that enzyme. By changing that shape of the enzyme, what the weed is doing is avoiding the full action of that herbicide. But the enzyme is still functional and allows that weed to survive. And so what we're doing as a lab is identifying these point mutations where that G turned into a C. As simple as that can lead to this resistance. And so what the lab test does, similar to that pigweed ID test that I talked about, it looks for that particular point mutation and it fluoresces a different color. And so we're able to detect that mutant or that point mutation that leads to the resistant genotype compared to the wild type or kind of that normal version that can be considered susceptible. But of course, the greenhouse trials are still very important because those capture both the non-target site or NTS and the target site mechanisms. But quickly, if you kind of go through the greenhouse, it's a demanding resource. So it's resource intensive. There's a lot of grow outs that need to happen across the various weed species and the populations. And so it's challenging to do a broad scale survey when you're restricted to just greenhouse work. That's where genetic testing comes in. So genetic tests are fast and high throughput. So really we only need a leaf sample to do these point mutation tests. We can do it in 96 well plates, so we can simultaneously test 96 plants in a given sample, but it is limited to these target site point mutations. And so we tend to have to know what we're looking for, and it is also expensive still. All right, and that's why we have this great partnership with NDSU and NDSU Extension, is that they're performing all of the greenhouse work, which is still very much needed, and then we're supplying also some genetic testing. The other benefit to having this relationship is that a lot of the greenhouse work is ongoing right now, right? It's going on during the off season when there's snow on the ground. With DNA testing, we can do that in season. So because we only need a leaf sample, you can do it at distinct points of time throughout the year. So whether it's before you plant, kind of what these arrows are indicating, before you plant, after an application, after harvest, you can test using genetic testing at any time. And so it kind of fills that gap that greenhouse trials cannot. So the goals of the survey are to seek these freaks, right? Seek these point mutations or these mutant plants. We want to make sure that it's easy collection and submission, quick reporting to any submitter that provides the samples, and then come up here in front of all of you and show you maps to show you the trends for these herbicide-resistant weeds. There's, of course, limitations. It's only known genetic markers, and these are linked to resistance. So we're not directly detecting the resistance. We cannot detect those new mutations and cannot detect non-target site mechanisms. So we have to be really careful with our language when we report to people. If we don't find the mutation, we cannot say it's a susceptible plant because there's other mechanisms that can lead to resistance. But this is the workflow. It's collect, test, identify, report, and map. We focus on two weeds, like I already mentioned, the pig weeds, mostly water hemp, and then kochia. Easy collection, we just need a leaf sample for this survey. We do not need the whole plant, but it is satisfying pulling out a whole water hemp. And sometimes when people start using our testing, they bring the whole plant to us. And what I tell them is just to take a cool picture of you with your kill, but I only need 1 or 2 leaves. That's all I need as a geneticist for the testing. So that's me attempting to look cool with my kill there. All right, so easy submission. All there is a submission form with 4 coin envelopes in a prepaid business envelope. So you just pack that all in, find a mailbox, and ship it to us. These are the markers that we are looking at associated with herbicide resistance. So we're looking at group 2, 9, and 14. In pigweeds, only the group 2 marker works across the species. With group 9 and group 14, those mechanisms are only found in water hemp and Palmer amaranth. With kochia, we're looking at all 3 of those resistant markers. And this is sponsored testing. And really the workflow, once we get one of those envelopes, hopefully packed with kochia or water hemp samples, it takes us just under 6 days to release the results to the client. We could be faster. The only reason why we're not is we're waiting for enough of those envelopes to come in to test because we're using a lot of plastic. We're a high throughput testing facility. So we're just kind of waiting for more samples to come in. Really, on a highly motivated day, we can get the results out the same day if needed. My tech probably would hate me to say that to you guys, but that's true. She rocks it out. So. But this is what a confidential report looks like. Essentially, if you have 3 samples, we're saying if we found the resistant marker based off of group 14, group 9, or group 2. And then we're saying resistant. If we didn't find the marker, we're saying wild type. Because as I said, we cannot confirm it's susceptible to the herbicide, only that we did not find that resistant marker. And like I said, this survey is sponsored through commodity groups. So we've gotten great support and funding through North Dakota Corn, North Dakota Soybean Councils. And recently, a minor donor was Northern Canola Growers. So we could take a peek into Minnesota as well using the survey. All right, I'm going to provide results in the last 10 minutes that I have here. For the state, I'm going to show you what we found last year and compare the prevalence to what we found this year. But then I got lazy and I'm only showing you last year's data for county and the individual level. So for pigweeds, we received samples from 23 counties and only 68 plants. We received 79 plants last year for pigweeds. We can further break that down into 33 water hemp samples and then 35 non-water hemp samples. Kochia, 36 counties and way more samples. Last year too, it was in the 400s, or 2024. So 2025 was 453 plants. All right, I'm going to show you the changes in the statewide resistant marker, kind of the prevalence or percentage in water hemp and kochia between 2024 and 2025. And so I have this broken down in group 2, 9, and 14, and then the horizontal axes there is showing you the percentage. So first we're going to start with the non-water hemp, or we're talking redroot, pigweed, probably Powell amaranth, those samples. We found 12% of that resistant marker in 2024 and 15% in 2025. So you'll notice these little arrows as we kind of do this comparison. The arrow is pointing to the 2025 survey result. So for kochia, we went from 24% to 32% last year. But in water hemp, we went from 48% having that resistant marker to 33% in 2025. We look at group 9 or glyphosate resistance. There was no change in water hemp. So 43% of the samples that came in were resistant or had that resistant marker. And then for kochia, we went from 58 to 59% having that resistant marker. And then group 14, water hemp, we went from 20% to 33% having the group 14 resistant marker. And then we saw an increase from 40% to 47% for kochia group 14 resistance. So this is the distribution of samples in 2025 that we received. So 79% of the counties we received samples from. So kochia only are the blue counties, pigweed only the yellow counties, both are the green counties, and then the number indicates the total number of weeds we received from those counties. So I'm going to show you kind of presence absence essentially of the resistant marker across the state according to county. So you'll see what group it is in the upper left-hand corner of each map. If the marker was detected, it'll be the colored county. If it was sampled but not detected, it's gray across all the maps. And then no sample, it's going to be a white county. So first, let's look at pigweeds. So group 2 counties that had the resistant marker are yellow, group 9 is orange, group 14 is red. And then what I did is aggregate, okay, which counties had all 3 of those markers? Those are the black counties in that bottom right map. And so for the group 2, that's including all pigweeds, but group 9 and group 14 are only water hemp samples. So only 3 of the counties were, I guess you could consider hot spots, meaning that they had all 3 resistant markers in them. Kochia, a different story. Just because we had a lot more samples, we're probably going to see a lot more resistance or the resistant marker. And so out of those 453 samples, group 2, group 9, and group 14 are quite widespread across the state. And of course, we found more counties that had all 3 resistant markers in them. So 22% of those counties out of the 36 contained all 3 resistant markers. So the cool thing about genetic testing is you can not only look at a single resistant marker, but you can test a single plant for all 3. So we can look at stacked resistances. And so that's these data here, just showing you the frequency of, for example, water hemp having none of the resistant markers that we test for, 1 of them, 2 of them, or all 3 of them. And so what we're seeing, at least in a comparison between water hemp and kochia, is that kochia has a higher frequency of 2 or more of these resistant markers when compared to water hemp. And just kind of a simpler way to say that is if we look at these plants, these individual plants that have 2 or more of these resistant markers, 24% of water hemp had that, but 42% of kochia had 2 or more of these resistant markers. So just to summarize, we saw an increased prevalence of all resistant markers when we compared the 2025 results to the 2024 results. 61% of the counties sampled had all 3 resistant markers, and 42% of the individual kochia had at least 2 of the resistant markers. When it came to water hemp and pigweed in general, there was only an increased prevalence of group 14 resistant marker from 2025 to 2024. Fewer counties sampled had all 3 resistant markers, and that probably just has something to do with fewer water hemp was sampled compared to kochia, and that 24% of water hemp had 2 or more resistant markers in them. But I think an important thing to point out is 4 out of every 5 weeds, regardless of the species that we tested, had at least 1 resistant marker. It's probably not surprising to anyone because we do have a herbicide resistant crisis. But that is something that's falling out of our genetic screening, is that there's a lot of resistance going on across the great state of North Dakota. So we do have some continuing projects. We really want to get green foxtail and wild oat online so I can share maps of those resistance as well. And so we're looking at group 1 and group 2 resistant markers there. We've already started to establish some markers to go after. These group 1 and group 2 resistant markers that I'm showing for green foxtail and wild oat, those are already confirmed in North Dakota populations based off of the resistance that NDSU greenhouse trials are finding. So we already have a basis of what mutations or point mutations to go after for these next tests. But I'm here because we also need your help. We did get funding confirmed through North Dakota corn and soybean for the continuation of the survey. So it really depends on you submitting samples to us. There will be collection kits across county extension offices. I am happy to mail you a bundle of collection kits if you want to reach out and request your own. I just hope that you return them to us with a bunch of weeds in those envelopes. And this, of course, is available across the borders too. All of our testing is available. It's sponsored testing in North Dakota. However, that's changing. I did get confirmation that Minnesota Wheat did approve some testing in Minnesota, which is great because with the canola project, we already confirmed water hemp has all 3 resistant markers, which isn't that surprising in Minnesota. We actually haven't confirmed ourselves personally with our genetic screening of that group 14 marker in Minnesota. It's probably there and we just haven't screened it yet, but stay tuned for that. And then water hemp is an important one for Montana, and so we want to start the survey over there. And we have confirmed the genotypes for group 14 resistance and kochia in Montana. So we suspect there's more counties that have that as well. So we're hoping to start this survey in both of our neighboring states this upcoming year. And with that, thank you so much for your time. That QR code brings you to our socials if you want to follow us. And I'm happy to entertain any of your questions. Thank you. Yeah, that was a great question. So for the audience, it was asking, do we have the ability to distinguish the monoecious pigweed? So like red root from Powell amaranth prior to doing the testing? And then whether or not there will be an assay or a test for group 14 resistance in those monoecious pigweeds. So the way that our pigweed ID test goes is it actually clumps all of those monoecious pigweeds together. So right now, currently, we don't have a test that can distinguish red root pigweed from Powell amaranth. However, we can sequence confirm that. It's just more work, more testing, and unfortunately more money for us to do that. And so if there's an interest in that, it's possible genetically. And I got the thumbs up from Jeff that there's an interest in that. When it comes to group 14 resistance, there's probably a couple markers that we can explore different than the one that we know with water hemp and palmer, and we just haven't looked at that in particular yet. But there is a marker out there that we could explore for those resistances in those monoecious pigweeds. The question online here is, when will foxtail screening be possible? Hopefully sooner than later. We kind of had to pause due to funding. Our funding periods just ended, and so we're able to pick that up as soon as new funding period has started. So we're not going to have it before the start of the cropping season this year, but hopefully by late summer, early fall, we'll have a better idea on it. But the great thing about this target site testing is that we're storing a lot of leaf samples. We got 147 samples through this survey last year that are wild oat and green foxtail, and we're going to be able to genotype or test those once the assay gets online. So we'll have a good idea of what was happening historically before we start officially rolling out the tests for future seasons. So.